In this lecture the students will learn about nuclear DNA and mitochondrial DNA. Human genetic identification for forensic purposes is achieved through the analysis of genetic profiles. A genetic profile or the genetic fingerprint of an individual is the description of a set of genomic loci that are specific to that individual. In accordance with international recommendations, currently, only genetic profiles obtained from autosomal short tandem repeats or STR should be used for genetic fingerprinting. However, in a considerable number of situations of human identification, autosomal DNA is highly degraded or is not available at all. In these cases the study of mitochondrial DNA for human identification can be the last appeal and by that reason has become routine. Ancient DNA specimens or samples that have been highly degraded often fail to produce results with nuclear DNA typing systems. However, recovery of DNA information from environmentally damaged DNA is sometimes possible with mitochondrial DNA. While a nuclear DNA test is usually more valuable, a mitochondrial DNA result is better than no result at all. The probability of obtaining a DNA typing result from mitochondrial DNA is higher than that of polymorphic markers found in nuclear DNA, particularly in cases where the amount of extracted DNA is very small, as in tissues such as bone, teeth, and hair. When remains are quite old or badly degraded, often bone, teeth, and hair are the only biological sources left from which to draw a sample. The primary characteristic that permits its recovery from degraded samples is the fact that mitochondrial DNA is present in cells at a much higher copy number than the nuclear DNA from which STRs are amplified. In short, though nuclear DNA contains much more information, there are only two copies of it in each cell, one maternal and one paternal, while mitochondrial DNA has a bit of useful genetic information times hundreds of copies per cell. With a higher copy number, some mitochondrial DNA molecules are more likely to survive than nuclear DNA. Mitochondria are cellular organelles that contain an extra chromosomal DNA, which is both different and separate from the nuclear DNA. Mitochondrial DNA makes up less than 1% of the total DNA content of a cell. Mitochondrial DNA has approximately 16,569 base pairs and possesses 37 genes that code for products used in the oxidative phosphorylation process or cellular energy production. There is also a 1122 BP control region that contains the origin of replication for one of the mitochondrial DNA strands but does not code for any gene products and is therefore referred to sometimes as the non-coding region. The total number of nucleotides in a mitochondrial DNA can vary due to small mutations that are either insertions or deletions. Most of the sequence variation between individuals is found in two specific segments of the control region, namely in the hypervariable region 1, HV1, positions 16,024 to 16,365, and in the hypervariable region 2, HV2, positions 73 to 340. Mitochondrial DNA presents several characteristics useful for forensic studies, especially related to the lack of recombination, to a high copy number, and to matrilineal inheritance. Mitochondrial DNA typing based on sequences of the control region or full genomic sequences analysis is used to analyze a variety of forensic samples such as old bones, teeth and hair, as well as other biological samples where the DNA content is low. Being a lineage genetic marker, mitochondrial DNA can provide information about ancestors, including health or disease information,